Good morning, students. Today's topic is low-level visual processing. So when the light enters into the eyeball, so the visual processing takes place in three levels. One is low level, intermediate level and high level. So we will be having three classes of this visual processing topic. So the objectives for today's class are photoreceptor cells in the eye and their properties. So we will discuss the various two types of photoreceptor cells and their properties and the mechanism of phototransduction and the physiology of light and dark adaptation, and lastly the photosensitive ganglion cells and their functions. So this is a flowchart depicting the mechanism of transmission of the vision. So first the visual impulse is taken by the photoreceptors which are present in the layers of retina, and the processing and transmission of visual impulse takes place in the retina. So this is the starting point, a low level of visual processing, and then the processing and transmission of visual impulse takes place in the visual pathway. So visual pathway is nothing but the starting of photoreceptors to the cortex. So like pain pathway or ascending pathways or tracts, we have the pathways also in special senses. For eye we have visual pathway, and for hearing we have auditory pathway, and for taste sensation we have taste pathway, and olfaction sensation we have olfactory pathway. Like this, for every sensation we have pathways. So that uh, intermediate, so this will come under intermediate level of visual processing. And then finally analysis of visual impulse and visual perception takes place in the visual cortex. Okay, so the three-part system hypothesis of visual perception is also the final result. That is how the person is able to receive the signals from a particular object or particular sight, the shape, contrast and the features of a particular shape. So that is called visual perception. So finally, this is the higher level of visual processing. So again the same thing, the mechanism of vision. We have three steps or three levels of visual processing. Initiation of vision, that is phototransduction mechanism, which is taking place in the rods and cones, and processing and transmission of visual sensation at retina and also visual pathway, and visual perception at the visual cortex and association cortex. So brief introduction about the retina, photoreceptors and pigments. So retina is the innermost layer of thin transparent membrane. So in the eyeball, so this is the structure of eyeball. So light and falls from here through the lens, and these inner layers are the first one will be the, this entire thing is the vitreous humor, and this is the sclera and the choroid layer, and then the retina. Okay, so here the cross section of the retina is taken from here, and then this is the optic nerve. So the optic disc is a circular pink color, only nerve fiber layers. So from here the optic nerve goes to the cortex, and macula lutea is yellow spot at the posterior pole temporal to the optic disc. So from the optic disc, the temporal side, macula lutea will be present, and fovea centralis is 1.5 mm, which is the most sensitive part of the retina, and ora serrata is the anterior serrated margin where the retina ends. Okay, so for understanding these terms, so once open the textbook and see the structure of the structural anatomy of the eyeball. So the layers of the retina are 10 layers. So this is the microscopic picture of the retinal layers. So if we see from inside, okay, so this is the outermost side. So this is finally the nerve going area. So the nerve will start from here and then goes into the optic disc and optic nerve. Okay, so the pigment epithelium layer, and then the rods and cones, and external limiting membrane, and outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglion cell layer, and nerve fiber layer, and finally the inner limiting membrane. So nerve fiber layer is completely made up of the nerves from the ganglion cells. Okay, so these 10 layers, names of the 10 layers you have to remember. Pigment epithelium and the photoreceptor layer, that is rods and cones, external limiting membrane, and outer nuclear layer, outer plexiform, inner nuclear, inner plexiform, and then ganglion cell layer, nerve fiber layer, and then inner limiting membrane. So this is again one more picture of the same layers of the retina. So pigmented layer, rods and cones, external limiting membrane, so that will be present here, and then outer nuclear layer, outer plexiform, inner plexiform, ganglion cell layer, and then the nerve fiber layer and internal limiting membrane. So fovea centralis, it is the most sensitive part of the retina, having maximum visual acuity. So it is the centralmost part of the macula lutea. Macula lutea is present at the posterior pole of the eyeball at temporal side of 1 to 2 mm diameter. So in the fovea centralis, why it is most sensitive means it is having only cones which are maximum in number and rods are absent. So here the cones are long with thin outer segment and reduced distortion as the layers push aside, and no capillaries are present here, and no convergence of cones, so one-to-one relay of cones with bipolar cells will be present. So here if you can see, there is convergence of bipolar cells onto the rod cells, and here there will be no convergence, so one-to-one relay of the cones with the bipolar cells are present. And if you see the differences between the rods and cones, the density and their distribution. So rods contain the pigment rhodopsin and cones contain the pigment iodopsin. And they are present 100 million per eye, and these cones are present only 3 million per eye. And these are helpful for night vision. The rods are helpful for night or scotopic vision, and cones are for day or color vision, photopic vision. And the rods are highly sensitive to low intensity light, and cones are less sensitive to low intensity light. Rods are having low visual acuity and cones are having high visual acuity. Rods are more numerous in the periphery, cones are concentrated in fovea centralis. And there is more convergence in the retinal pathway for rods, and there is less convergence in the retinal pathway for cones. So these are the pictures of the rods and the cones. So outer segment if you see, it is more large in rods and less small length in cones, and these are the discs and the connecting cilium. Inner segment containing the cells, that is mitochondria, and the nuclear part, axon, and then synaptic ending. The structure of rods, it is 40 to 60 micrometers long. The outer segment is cylindrical and highly refractile. It contains the pigment visual purple or rhodopsin, and it is lipid protein lamellar disc. Okay, so it is a disc. So name is there. So it is lipid protein which are arranged in lamellae. And the inner segment is thicker, and there are two regions, ellipsoid, myoid, and rod spherule. Okay, so in the inner segment we have two regions, ellipsoid and myoid, and rod spherule are present in the inner segment. And cones 40 to 80 micrometers long. The outer segment is conical and contains the pigment iodopsin, and inner segment again two regions, ellipsoid and myoid, and contains cone pedicle. So visual pigments or photopigments, there are actually four different photosensitive components present in the retina. One in the rods, rhodopsin, and then other in the three in the cones. Okay, so three types of cone pigments are present which are sensitive for three different colors: red, blue, and green. And the actual light sensitive pigment is the retinine, and opsin is an integral membrane protein. Okay, so retinine is the chromophore molecule which is same in all the photopigments, but opsin difference in each of the four pigments. Okay, rhodopsin is the visual purple, protein opsin or scotopsin, and carotenoid retinol as an aldehyde of vitamin A. And the molecular weight for the rhodopsin is 40,000, and it absorbs primarily yellow wavelength light, transmitting violet and red to appear purple, so-called visual purple. And the cone pigments, three types, which respond to specific wavelength of light. So because of this specific wavelength, they are categorized into three: red, blue and green. Chromophore, that is the retinal part, it is 11 cis retinol, which is same for both, for three pigments, and difference in only opsin portion, which is the integral part of the membrane. So if we see the light induced changes in the eyeball, when light strikes the eye, a hyperpolarizing potential is generated in the rods and cones known as photoreceptor potential. So the conversion of light energy to neural signal by visual pigments in the photoreceptor is known as phototransduction. And the three steps in the light induced changes in the uh, rods and cones are rhodopsin bleaching, rhodopsin regeneration, and visual cycle. So mainly the things take place in the rods. So coming to the rhodopsin bleaching and regeneration. So bleaching means it's utilized in the phototransduction process, and regeneration is it is produced again, or recycled again, so that the normal rhodopsin comes back. So when there is no light in dark, the rhodopsin remains in a less stable form, which is 11 cis retinol. So this is the less stable form. When light strikes the eye, it absorbs a photon. So rhodopsin absorbs a photon and gets isomerized to all trans retinol, which is a more stable form. All trans retinol will be a more stable form. So this isomerization induces the opsin to undergo a series of rapid conformational changes and converts rhodopsin to meta-rhodopsin 2. So this is when light energy strikes the rhodopsin, it will be transformed to all-trans retinol, and this all-trans retinol, which is more stable form, will be converted to meta-rhodopsin 2. Final is meta-rhodopsin by a series of changes, that is batho-rhodopsin, lumirhodopsin, meta-rhodopsin 1, and then finally meta-rhodopsin 2. So this meta-rhodopsin 2, which in turn activates the G protein, and finally the process of uh, phototransduction or chemical reactions will take place. So what is the rhodopsin bleaching means? The all trans retinol is separated from the opsin after isomerization. As a result, reddish color imparted to opsin by the photopigment disappears and opsin becomes pale yellow. Okay, so that process is called bleaching. So after isomerization, the all trans retinol will be separated from opsin. Okay, so that will be imparting reddish color to opsin by the photopigment, and that red color will disappear and opsin becomes pale yellow. So because of that yellow color, it is called rhodopsin bleaching. And then the all trans retinol moves to the pigment epithelium, where the enzyme retinal isomerase converts it into 11 cis retinol. Okay, isomerase will converts it into 11 cis retinol in the retinal pigment layer. So the photopigment then gets translocated to the outer segment of the rods to recombine with the opsin, replenishing the rhodopsin store. Okay. So this 11 cis retinol will be combining with opsin in the outer pigment layer, outer segment of the rods, to replenish the rhodopsin stores. So this is called regeneration of rhodopsin. So it takes few minutes for one regeneration cycle to get completed. Okay, so in few minutes the 11 cis retinol will be combining with opsin to form the rhodopsin in the outer pigment or outer segment layer of the rods. So this is the when light strikes the retina, the rhodopsin will be converted to all trans retinol, that is the most stable form, and the rhodopsin will be converted finally to meta-rhodopsin by a series of changes, and this will be the rhodopsin bleaching, that is the color imparted. red color will be converted to yellow color by changing of rhodopsin to all trans retinol, so that is called rhodopsin bleaching. And then this is again converted to 11-cis retinol by isomerization, so which is called rhodopsin regeneration. So the visual cycle. The cycle starts with all trans retinol, which is the released from the rhodopsin. By bleaching, the all trans retinol is released. So that will be converted to all trans retinol, and some part of the all trans retinol will also come from the systemic blood circulation. So this is otherwise the vitamin A, and this will be taking place in the outer segment, and this all trans retinol will enter into the retinal pigment epithelium, where the retinol is used to generate these retinal esters with the help of this RP, retinal pigment epithelium 65 gene, and this takes place in the retinal pigment epithelium. And finally 11 cis retinol will be generated, and that will be again by oxidization converted to 11 cis retinol, and this is taken back into the photoreceptor layer to regenerate the photosensitive pigment, that is rhodopsin. So cis retinol combines with opsin to form the rhodopsin. So this is the actual visual cycle, and rhodopsin, you can see here, it will be producing the meta rhodopsin 2, which is helpful in the actual transduction mechanism by producing the transducin. And this is again converted to opsin, apoopsin, and apoopsin along with when combined with aldehyde 11-cis retinol with opsin will be producing rhodopsin. So now we will see the actual process of phototransduction, which is nothing but the conversion of light energy to nerve impulse. Okay, so here the series of changes taking place, starting with induction or entry of the light rays into the retina. So the ion channels will be converted, so causing the entry of sodium and calcium into the inside of the cell, and it involves the conversion or the utilization of cyclic GMP. So with the help of GTP, conversion to GMP, and then this is the step 5. So in one more, so this is the actual mechanism of phototransduction. In this flowchart you can see, so when light falls on the eye, so there will be conversion of the 11-cis retinol to all trans retinol, conformational change in the opsin forming meta-rhodopsin 2. So this meta-rhodopsin 2, actually I said you in the previous slide, so this helps in the series of transformations taking place, so that the sodium entry takes place. Activation of G protein transducin. So this is the G protein transducin by the meta-rhodopsin 2. And then activation of phosphodiesterase that converts cyclic GMP to 5-GMP, and then decreased cytoplasmic level of cyclic GMP leading to closure of cyclic GMP gated sodium channels, and decreased sodium influx causing hyperpolarization, decreased release of synaptic transmitters, and then response in the bipolar cells. Okay, so this cyclic GMP levels decreased means this helps in the closure of the sodium gated channels. So normally there is sodium influx, so that will be decreased because of hyperpolarization. So there is decreased release of synaptic transmitters, so that will cause a response in bipolar cells. So the receptor potential is when light strikes the eye, hyperpolarizing potential generated in the rods and cones is known as photoreceptor potential. So in the cones and rods, so how this receptor potential differs means, the cone receptor potential has sharp onset and offset. It comes in sharp time, and then again it goes off in a very small time. And rod receptor potential has sharp onset, that is, it also comes very in short time gap, but it goes off very slowly. And rod responses are proportionate to stimulus intensity at illumination levels below threshold for cones, and rods detect absolute illumination. And cone responses are proportionate to stimulus intensity at high levels of illumination when rod responses are maximal. And this generate response to change in light intensity above the background. Okay, so that's why cone responses are responsible for the color vision. And this is again one more picture, the transmission or the phototransduction when in darkness and then in light. The changes which are taking place in darkness and also in the light. In the darkness, what is happening? Cyclic GMP gated sodium channels are open. Inflow of sodium, that is dark current taking place, and then the membrane potential is taken back to minus 30 millivolts. So membrane potential is decreased so that the glutamate is released at the synaptic terminals, which inhibits the bipolar cells. So the transmission will be decreased in darkness. So this is the process taking place in the darkness. So when light is not present, the sodium influx will be present, but it is decreased so that the membrane potential is decreased to minus 30 millivolts and glutamate will be released at the synaptic terminals. And when there is light, isomerization of the retinal activates enzyme that breaks down the cyclic GMP. So the cyclic GMP channels will be closed, so there will be no influx of sodium. So there will be hyperpolarizing receptor potential. So here there will be depolarization. The membrane potential decreases to minus 30, and here there will be, because of sodium influx of stoppages or slowing down, there will be hyperpolarizing receptor potential. So that will be causing release of glutamate to be turned off, or there will be no release of glutamate, which excites the bipolar cells, and there will be the signal transmission taking place through the optic nerve back to the visual cortex. So this is standing potential or dark current which is taking place in the darkness. Okay, so this is the production of cyclic GMP, and there is sodium gates will be open. There will be sodium influx taking place leading to depolarization. And when there is light, hyperpolarizing receptor potential takes place because of decreased cyclic GMP or the closure of sodium gated channels, so that the sodium influx does not take place and there will be hyperpolarizing potential and glutamate will not be released. So this is this picture and the previous picture and that before picture, flow chart picture. So all these pictures you should remember, and the sequence of events, what is happening in the darkness and what is happening in the when there is light. Okay. So after learning the process or mechanism of phototransduction, we will see the processing and transmission of impulse in the retina. So what is for this you have to know the receptive field, that is an influence area of a sensory neuron which is circular in configuration. So the receptive field of individual photoreceptor for in darkness the photoreceptors get depolarized, neurotransmitter glutamate is released, and in light photoreceptors get hyperpolarized and less neurotransmitter or no neurotransmitter is released. So this is the mechanism of phototransduction. So the receptive field is the influence area of a sensory neuron which is circular in configuration, which helps in the receiving of visual impulse from the retina. So we have various types of cells: horizontal, bipolar, amacrine and ganglion cells in the retinal layers. So the receptive field of these individual cells: in horizontal we have large receptive field. Its function is to enhance visual contrast by lateral inhibition, and bipolar cells are of two types, one stimulated and other inhibited by photoreceptors. It provides second mechanism for lateral inhibition. And amacrine cells, they help in processing of spatial information. And ganglion cells, their mechanism is centers around antagonism, so that finally the ganglion cells, only from the ganglion cells only the nerve fiber emerges out into the optic nerve. So if we see the horizontal cells, so each and every cells, what are the physiological activities taking place? So here in the horizontal cells, as we have seen in the previous slide, it is having large receptive field. So there will be phenomenon of lateral inhibition. That is lateral inhibition, if you remember from the sensory encoding class. So the surrounding will be inhibited and the center will be highlighted. So that is lateral inhibition. So here the horizontal transmission of signals in the outer plexiform layer will be present. So minute spot of light, that is the centralmost area, will be excited and the surrounding area will be inhibited. So that is the peripheral bipolar cells will be inhibited. So this enhances the visual contrast. And the concept of receptive field, so again this is the influence of sensory neuron. So it is very large compared to the photoreceptor cells. And the bipolar cells are two types. So these are the first order neurons of visual pathway. So some bipolar cells depolarize while some bipolar cells hyperpolarize. Okay, so here you can see bipolar cells provide opposing excitatory and inhibitory signals. So depolarizing cells respond to glutamate and some are directly stimulated by the photoreceptors leading to excitatory, and others are indirectly inhibited by the horizontal cells. So horizontal cells will be inhibiting some bipolar cells, so that is the lateral inhibition, so that the center part will be activated. Receptive field of the bipolar cells has got a center surround antagonism. Okay, so what is the center surround antagonism? We will see in the coming slides. So this is the picture of that center surround antagonism. So center will be activated, surround will be inhibited. So here center will be inhibited, surround will be activated. So this mechanism is mainly to increase the visual acuity. So this is one picture where the center cells are being inhibited and the peripheral cells will be activated. So minus means inhibition. So center surround antagonism is a mechanism to increase. So how this center surround antagonism takes place means, in the center, suppose there is a cone which is depolarized cone, so there will be release of glutamate. So this released glutamate in the on center bipolar cell, the hyperpolarization takes place and the transmitter release is decreased and there is firing on center ganglion cell decrease. And whereas off center bipolar cell, okay, this will be depolarized and transmitter release increase, so there will be firing of off center ganglion cell increases. Okay, so from the rods and cones here, from the center surround antagonism, that is the cones which are depolarized and releasing glutamate, so these bipolar cells are also, one will be hyperpolarized and one will be depolarized. And here also same, when the cone is hyperpolarized, glutamate release is decreased. So one will be depolarized and one will be hyperpolarized. So when there is depolarization, firing increases. When there is hyperpolarization of bipolar cell, firing of the ganglion cells will be decreased. So one more type of cells are the amacrine cells. You can see here in the picture, bipolar cell is present and this green colored one is the amacrine cell and this is the ganglion cell. So they receive information at the synapse of bipolar cell axon with the ganglion cell dendrites. Okay, so the temporal processing at the other end of bipolar cells. Electrically produce depolarizing potentials and spikes which act as generator potentials in the ganglion cells. So these amacrine cells help in producing the depolarizing potentials and spikes which act as generator potentials in the ganglion cells, help in temporal summation and initial analysis of visual signals. So these amacrine cells help in temporal summation of the signals and also initial analysis of the visual. And the next ones are the ganglion cells. Electrical response of bipolar cells, which is modified by amacrine cells, and finally reaching the ganglion cells, producing the action potential and sending the signals to brain. On and off center ganglion cells. These produce propagated spikes. On center increase their discharge, whereas off center decrease their discharge. Okay, so on center means positive, stimulated, so that will be causing increased discharge, and off center means negative or inhibited, so that will be having decreasing discharge. So depending upon their function, there are W, X and Y type of ganglion cells are present. So what are these W? So W type of ganglion cells are small, less than 10 microns diameter, and X are medium, 10 to 15 diameter, and large. Y cells are large, diameter up to 35. So out of these three, the numerous ones are the X type of cells, and W type of cells are 40%, and lesser are Y type of cells. And the W ganglion cells, dendrites, are widely spread in the pigment layer, and X are the having small receptive fields. Dendrites do not spread, and Y type of cells have very broad dendritic field. And W pick up signals from the rods, and X pick up signals from at least one cone, and Y pick up signals from widespread retinal area. So W are responsible for rod vision in dark and detect directional movements, and X are responsible for color vision, and Y respond to rapid change in visual image. So X are sustained cells and Y are transient cells. So in this picture we can see the difference in center surround antagonism when there is spot of light and bar of light. So when there is bar of light across the both center and surround, the region will be less effective. Okay, so one will be on and one will be off, but both are less effective at stimulating the ganglion cell. So here when there is spot of light, so in the center on center field, so the center part will be on and surround part will be off, and in off center field, so central part will be off and surrounding part will be on. So this is the different cell layers. So one more picture which is showing the direction of light, and then the pigmented epithelium rods and cones which are present in the last layers of the retina. And now we will see the visual adaptation, that is in presence of light and in presence of dark, how the human eye will be adapted. The human eye has ability to discriminate and identify objects under a wide range of illumination. But when light intensity changes, the visual system requires some time for adaptation of eye to respond optimally. That is, suppose we have been present in the light and suddenly we have entered the dark. So the intensity changes. So visual system requires some time for adaptation of eye. Opening and closing of pupil adjusts the amount of light entering the eye. So this is one mechanism of adaptation. And neural adaptation also present, which helps in signal transmission by bipolar cells, horizontal, amacrine and ganglion cells. So if we see the importance of these adaptations, so these help in detection of images on the retina, which is a function of discriminating between dark and light spots. So for example, when entering a sun from movie theater, even the dark spots appear bright, leaving a little contrast. And if we enter darkness from light, light spots take time to register on retina. And in light conditions, most of the rhodopsin has been reduced to retinol, so the level of photosensitive chemicals is low. And in dark conditions, retinol and opsins in the rods and cones are converted back into light sensitive pigments. So therefore, sensitivity of retinol automatically adjusts to the light level. So if we see the dark adaptation, it is the ability of eye to adapt due to decreased illumination. The time taken to adapt is called dark adaptation time. So rods use more in dim light scotopic vision than cones. So rods are used more in this dark adaptation. And also in dim light, the pupil dilates more to allow maximum possible light to enter through the eye to have optimal vision. So these are the graphs of dark adaptation of rods and cones. So first, initially, as the time we are present in darkness. So when we enter from lightness into the light area to dark area, first the cones are being adapted and then the rods are being adapted. So these are the dark adaptation curves. So here the graph shows visual threshold falls progressively in dark room for half hour until relatively constant value is reached. Okay, so the threshold value will be falling in the dark room for half an hour. Okay, so the time in the dark room, the threshold falls for half an hour until the constant value is reached. And then this graph shows increase in sensitivity of retina in two steps. So first, rapid, short and small due to cone adaptation. So this is rapid fall in the intensity or threshold, rapid, short and small due to cone adaptation, and second one slow, long and large due to rods adaptation. So break point is cone and rod breakpoint or alpha point. Here, after the cones being adapted, and then the rods will be being slowly adapted. So this point of contact between the rod curve and cone curve is the break point. So the mechanisms of dark adaptation are visual pigment mechanism and also neural mechanism. So in visual pigment mechanism, there is reversal of mechanism of light adaptation. That is, there will be regeneration of visual pigment. So in the phototransduction mechanism, we have seen so retina, rhodopsin, bleaching and regeneration. So in darkness, what happens means reversal of mechanism of light adaptation or phototransduction mechanism. So there will be regeneration of visual or rhodopsin pigments and the change in pupillary size, that is dilatation to 30 times. And if we see the neural mechanism, there is visual change in retina. That is cells that is horizontal, bipolar, amacrine and dual ganglion cells. So because of that neural mechanism, the transduction or the transmission takes place to the optic nerve. Okay, in vitamin D deficiency, there is elevated threshold for dark adaptation due to depletion of photosensitive pigments. So this leads to night blindness or nyctalopia. And next, if we see the light adaptation, it is defined as the process by which retina adapts itself to bright light. So it is very quick and over in 5 minutes. It is merely disappearance of dark adaptation. So it takes about 5 minutes and immediate reaction to sudden bright light, pupil constricts and amount of light entering the retina is reduced. And if we see the mechanisms for this light adaptation, so neural adjustment is very transient effect. So the sensitivity of retinal photoreceptors decreases, that manifests in the form of decreased burst of activity to a constant stimulus. Okay, so the horizontal cells may have feedback inhibitory effect upon the photoreceptors. So the inhibitory effect will be present, so the glutamate will be reduced. And the visual pigment mechanism, there will be reduction of rhodopsin and cone pigment due to bleaching in the light. Okay, so the rhodopsin converted to all trans retinol and opsin. So rod responsiveness decreases and rhodopsin becomes less available. So as the rods fail to respond further to bright light, cones come into play. So that with increasing light intensity, rods become deactivated and cones get stimulated. And there will be pupillary constriction. Okay, so that's all for today's class. I hope you understood at least some part of the class, because the mechanism of phototransduction is okay, but that centers around antagonism and the various types of cells present in the retinal layers. So that is little confusing. So read from the textbook twice and then try to explain on your own and try to make notes on your own, so that you will have a better idea. So for today's assignment, draw the picture of or flowchart and picture of the mechanism of phototransduction and also dark and light adaptation mechanisms. Okay, so both these questions are the today's assignment. Thank you all.